Thank you all for being here. And we have the pleasure to have with us Professor Michalis Aivaliotis. Professor Aivaliotis is coming from the Aristotle University of Thessaloniki, and he's a Greek expert on omics and mass spectrometry, so we do have this expertise here in developing, and he's going to present us his talk on on clinical proteomics in the era of precision medicine. Thank you.
So welcome everybody. I'm very happy to be here and honored for this invitation. Thank you, Christos. It's a great opportunity to have mass spectrometry with clinical laboratories and clinical chemistry. So I'm going to share some thoughts, some information about how clinical proteomics is actually trying to go into the clinical practice in the era of precision medicine. So first of all, I would like to highlight that the Proteomics Society is huge, have many fields, and from the beginning, almost 40 years ago, one of the major directions was clinical proteomics. And this is a recent publication, actually it's a special issue in the top proteomics journal, Molecular Cell Proteomics, where a promise becoming reality. All the scientists are trying to make the clinical proteomics an actual part of the clinical practice. So before start with the details, it's very important to realize that we are facing precision medicine as a requirement and personalized medicine. Daily medical practice meets many limitations and many gaps in knowledge, and that's a problem in the application of health. And the problem is that we don't fully understand human beings and how the human beings are affected from the environment. So precision medicine is trying to collect all the information, including molecular information in detail, in order to provide precise medical support and personalized medicine, which is really difficult to do it, but at least they are trying to do it. So regarding human beings are complex biological systems, so for sure we don't have a full description of a disease with one biomarker or two biomarkers or three biomarkers, or we cannot have treatment with a few drugs available. We need to fully understand the details of the human body, combining different levels of information from the molecular networks and all these molecular omics to the cellular networks and the organs and tissues. So today I'm going to focus mostly on proteomics, but in reality, we need to integrate all this information. We have to consider also that we have a heterogeneous system. So in order to have a precise and personalized medicine, we need to clarify what are the differences between the different individuals and what are the differences on physiological and pathological phenotypes. In order to do that, you have to collect all this individual information on a molecular level to combine them and try to understand the system. And the system is not only one. In biological in human body, we have many different biological systems. So at the end of the day, we need to find the interactions, how do they work, interacting each other, and how these interactions affect the biological system and the phenotype. So we have tools today, first of all, instruments. Without them, we cannot collect all this high-quality data, and we can collect individually every level of molecular information. We have to do that because we have technical limitations. We cannot have one sample analyzed by one experiment and have all the information there. So we have to separate the questions, we have to separate the techniques, we have to separate the molecular levels. But at the end of the day, we need to integrate all this information again by collecting, first of all, the information, integrate them in multi-omics or transomics platforms, and try to see how the system works when you have all together in the system. And that also somehow defines how modern molecular diagnostics should be. We need to have panels of multi-levels of molecular information in order to better describe a disease or a physiological condition. So we need DNA, RNA, proteins, and metabolites. And proteins and metabolites are closer to the actual phenotype because are closer to the biochemical procedures, and they have also the highest dynamic in comparison to DNA. Nevertheless, nowadays, non-coding RNAs are becoming very popular because they show similar dynamics as proteins and metabolites. So Nobel Prize this year was for the discovery and study of non-coding RNAs. So the future also of this kind of molecules will be very promising. In order to have, of course, all this information together, we need tools. And again, bioinformatics is giving us solutions for integrating all this data. It's still complex for most of us and even more complex for medical doctors and clinical experts. And we need to find ways to visualize this data without confusing people or make people not looking at them. Another parameter which is very crucial is to understand that we have all these microenvironments. So we have to see how the different molecules in different microenvironments are working, are functioning, and also to find the differences between the microenvironments and how the microenvironments affect the molecular networks. Another very important information is that the procedure from the gene to the final protein product is not straightforward. We have many levels that modify the final product, and from a set of 20 to 25,000 genes, we can have more than 1 million proteins. And these are the actual biochemical players in the cells and the biological systems. So the question today is not which gene under which conditions is responsible for a disease or a physiological condition, but which of these 1 million possibilities of proteoforms is actually one of the players of physiological or pathological conditions. So we have to find the combinations of different proteoforms that define the phenotype that we see, physiological and pathological. So that is partially made of all these chemical modifications that enzymatically happen on proteins after their translation, after their synthesis. So the system prepares all the proteins in order to be functional for specific reasons, for specific conditions, and all these modifications are also putative biomarkers of a specific phenotype. But also, all this complexity affects our capabilities in clinical practice. So we have to first build all this knowledge, build the tools, and then try to build the applications for the medical practice. In order to do that, we have to zoom in and zoom out in the system. So zooming in on protein isoforms, individual proteins, we can get details for these proteins which are required, like structure, regulatory sites, dynamics, and so forth, so on. But we cannot find, by these individual studies, the dynamics of each protein in the system. So we have to zoom out and see, using proteomics, what are the correlations between the different proteoforms and proteins in each condition. The main tools that we have today in proteomics are, the major tools are three. So we have mass spectrometry, Nobel Prize in 2002 for the capacity of mass spectrometry to measure proteins and do proteomics today. Protein biochemistry has to change a little bit in order to be compatible with the mass spectrometry, and of course, bioinformatics to deal with the complexity and the big amount of data that we produce from this analysis. But we have to think always that this is a multidisciplinary field, and we have to combine these three pillars with other methods as well. So the typical proteomics are, let's say, separated into two approaches, the bottom-up and then the top-down. So the bottom-up extracts all the proteins and digests them using a very specific enzyme into peptides. And we do that because our tools, liquid chromatography, mass spectrometry, is better analyzing peptides rather than proteins. So this very important step for doing proteomics is giving us many advantages, but also has one major limitation. By this digestion, we lose the information about the proteoforms. So we cannot separate which proteoform is responsible for what. We can identify proteins, characterize them, but we cannot separate them into proteoforms. In order to deal with this problem, the last 20 years, scientists are trying to apply top-down proteomics. Instead of digesting proteins, they try to analyze proteins, induct proteins, proteoforms into mass spectrometers. we have limitations here because the current technologies in liquid chromatography and the current technologies in mass spectrometry cannot deal with thousands of proteins, which are huge, heterogeneous, and we are limited nowadays in the analysis of small sets of proteins. So we cannot do actually proteomics with top-down, but maybe in the future we will be able to do that. So the typical protocol starts from the system, isolation of the cell or tissues, proteins. Proteins are digested into peptides. We have the liquid chromatography always coupled with mass spectrometry. It's very important here to clarify that there are special systems for proteomics. They are not the same as metabolomics. So we have to use special instruments here. And then we need to know the theoretical proteome of the system, the biological system. We need to know the genome of the organism. And then we do identification of peptides, assemble of the peptides into proteins, and I have a list of reliable, reproducible identification of proteins. And then I can do the functional analysis of all this information in relation to our initial biological question. This is a typical protocol. Nowadays, we have different kinds of mass spectrometers like this one or this one. In all cases, are very expensive hybrid mass analyzers with demands regarding the usage of them. So the personnel needs to be well-trained and has an expertise. And we have different ways to detect the signals of peptides and proteins. So one approach is to separate all the peptides and one by one separating and analyzing through a SMS analysis for the sequencing of the amino acid sequence of each peptide. This is one approach. And the other approach is, instead of analyzing separately each peptide, to have a pool of peptides, fragment them all together and have a mixed spectrum of MSMS, and then bioinformatics are used in order to find which fragments correspond to which peptide and have the sequencing of each peptide. So the reason for these two applications, I'm going to show you in the next slide, but before that, this application is the discovery approach. So I'm trying to analyze everything, to detect everything, and find every peptide and protein in my sample, in case that I would like to discover something. But if I have already information about peptides and proteins, then I can have a list of previously analyzed peptides and proteins from different databases with real data, and then I train the mass analyzer to find these signals like an antibody, but instead of one antibody for one protein, I have thousands of signals for thousands of proteins. So in 1 hour or 3 hours, I can see simultaneously, quantitatively, thousands of proteins in high resolution and high sensitivity. This is the targeted mass analyzer and the targeted approach, and this is actually the application of proteomics that is closer to go into the clinical practice because of this targeted approach and the ability to provide quantities. So the limitations of the methods. No, there is no perfect method. Always you have to combine methods and then integrate the results. So the first method, you see here the M/Z of peptides purified during the analysis and the retention time, the elution of them out of the liquid chromatography, and these red boxes indicate which signals, which are these yellow-green signals. Each yellow-green dot here is a peptide. So you see that the software and the mass analyzer can only identify some of them, not all of them. For example, here I have some gaps, and this is one of the major limitations. I cannot see everything in my sample. Always I'm losing part of the information. This is for every method, for every experiment. So I can either simplify my complex mixture, make it simpler, less proteins, less signals, or make a faster mass analyzer. Of course, the companies are trying to do that, but this is more difficult. In most of the cases, you try to simplify your signal, your sample. In another approach, you try to analyze everything, but you do that by pulling together different peptides. So the MSMS spectra for the sequence of peptides are mixed spectra from different peptides. And then you do bioinformatic analysis to identify these signals. And that makes a high probability of false positives and negatives. So nobody is perfect. And this is the target analysis where I ignore all the other signals and I only analyze the signals that I know that are related to my initial question. And of course, bioinformatics in each step. So I have many steps of bioinformatic analysis from the acquisition of the sample, statistic analysis, quality control, so forth, so on. I'm not going to bother you today with bioinformatics, but in case that you would like in the future to invest in this field, you have to know that bioinformatics of proteomics is 1/3 of the whole effort. So you have the preparation of the sample, the mass spectrometry analysis, and the bioinformatic analysis. 1 week measurements in the mass analyzer, months or years of analysis of the data in order to extract the meaningful information. So proteomics readout are multidimensional. You can do comparisons of global proteomics. You can see interactions. You can see post-translational modifications. You can have single-cell proteomics, and all of them, you can apply them in clinical samples. And all of these approaches are needed in clinical practice because they provide information that can be used as biomarkers for any case, but also they provide information in order to understand the system and better define what are the biomarkers for staging, differentiation, and so forth, so on. An example here of a complex disease, neurodegenerative diseases. So if somebody would like to study neurodegenerative diseases in order to gain insights, but also find some meaningful biomarkers or drug targets, first of all, he or she has to decide what kind of proteomics will apply. In which part of the system these proteomics will be applied, because a system in human body has many topologies, many cells. So in which of them I will ask a question and I will do the analysis. And then in which conditions, in which time points I'm going to apply this. If I apply a question and select clinical data and clinical samples for the analysis in the wrong timing or in the wrong condition, all the information that I will get maybe will not be so accurate. So I have to decide what are the proper conditions, what are the proper time points. And then I can address different kinds of questions. What are the protein-protein interactions affected in each case? Do I have aggregations? And if I have some aggregations in my biological system, which kind of aggregation is this? Which protein is involved and how is developed? Do I have protein nucleic acid interactions? Are they regulatory, are normal, are physiological, pathological? What is the effect of these interactions in the system? The same starts for other kinds of interactions like protein lipids, and of course I have protein-protein interactions. In the case of modifications, all these are parameters that may affect my result and I need to consider. For example, stability, activity, regulation of the protein, binding of the protein with other partners, protein turnover, proteolysis, localization, the existence of multiple PTMs, post-translational modifications. What is the relation of the different modifications? For example, in some cases, you need to combine different post-translational modifications in order to have the activation of one event, and all these are important biomarkers of the development of one condition. And of course, you can use also the same method for drug discovery and drug development. So one example of biomarker discovery is like that. So you start with a small sampling, small samples here, small amount of samples. You do experiments in clinical samples, but also in model systems. You gain information about this system by the analysis, and you discover many, many biomarkers at the beginning, but slowly you go through this procedure and you try to identify a small set of proteins, and you apply this small set of proteins on a bigger cohort of samples and conditions in order to validate them. And of course, when you have ready all this information, you need to have access in databases. You need to integrate the clinical data in these databases where you need bioinformatic algorithms that will find what is meaningful from this analysis and help the clinician not to spend time to understand this information, but rather have a decision out of this analysis, which is also not available yet. One example in a recent publication indicated highlighted that they made an analysis of the human plasma proteome in 4 hours using mass spectrometry pipeline, a standard operation procedure from real samples, and using algorithms that were developed in their lab. In 2.5 days, they had a treatment recommendation for the doctors. So they could have diagnosis, and they could have at the same time recommendation of a putative treatment. Of course, this is not yet applied in real clinical practice. It's just a case where we try to see what are the capabilities of this application. So another way to do this validation of clinical proteomics results is to go through the discovery phase, where you analyze real samples using standard operation procedures, and the outcome of this analysis is a list of biomarker identifications. Then normally we do biomarker verification using other methods, in most of the cases immunoassays, and then we try to understand why these biomarkers are increased or decreased or differentiated, and correlate this with the initial clinical question. This is the translational phase, and this is the discovery phase of this clinical proteomics application. And when we try to find reasons why clinical proteomics is not yet in the clinic, as you can see here, you see here some of the reasons. It's very expensive. It has low throughput at the moment, and it's not easy to analyze the data. And of course, the sample preparation, it's much more complex. So these are some of the challenges in order to make clinical proteomics using mass spectrometry to go closer to the clinical application. Whereas immunoassays are in the clinic already, they cannot discover new things easily because they have biased analysis of specific targets. They have low cost, but they have low multiplexing capabilities. They are high throughput, they have high specificity, high sensitivity. They have slower assay development because we don't have always the right antibodies. And it's very easy to do data analysis and very simple sample preparation. Of course, there are some other ways to see this translation from the discovery to the clinical application, and these are some of the limitations that you face in each step. So you go through this procedure, and in each step you have to overcome some limitations. I'm going to go into details. So a very simple example from our research in the lab. I have to admit that I'm doing preclinical proteomics. So this is an application of serum proteomics analysis in cancer patients and normal individuals. You see, we could identify from serum 3,000 proteins, which is a huge number. If you consider that the standard blood proteins are around 1,000, all the others are some isoforms or proteins that were secreted from cells. You see that we have major differences between the cancer and the physiological phenotype, but also we have a high similarity. A principal component analysis for the pattern of each individual could discriminate. These are the patients, and these are the different patients, and this analysis considered the proteomic profile of the serum of each individual, and these are the healthy. So with this analysis of the 3,000 proteins, we could discriminate by the different profiles the healthy, and the diseased cancer patients. This is a heat map of all the proteins identified. So you see that these are the normal individuals, and these are the cancer patients. And you see that in this region, these are proteins here. In every row, you have one protein identified. You see that in this region, you have many proteins overexpressed, upregulated in higher abundance in the cancer patients. And I think that this is the pool where we are going to search for some biomarkers. And of course, we could see which proteins were increased in cancer patients and which proteins were decreased. And from this pool, we're going to do validation using immunoassays. Another example is CSF proteomics, another biological fluid, which is quite popular for multiple sclerosis patients. 3 conditions: control, preliminary, let's say, a starting condition before the multiple sclerosis, and the multiple sclerosis patients. So you see that after the analysis, it wasn't so easy to discriminate between the different individuals, but we could find characteristic proteins for each comparison: multiple sclerosis versus control, healthy individuals, multiple sclerosis with a syndrome which is before the multiple sclerosis development, and the syndrome with healthy individuals. All these proteins, I think, we could be further validated with immunoassays. Now, the future of proteomics, please remember this name. This is Matthias Mann. It's one of the pioneers in proteomics. I think that maybe in the future we'll get a Nobel Prize. It was actually the postdoc of one Nobel Prize laureate in 2002. John Fenn took the Nobel Prize for making mass spectrometry able to analyze proteomics. And this guy, the last 10 years, has focused his research on clinical proteomics. And one of these papers, called Unbiased Spatial Proteomics with Single-Cell Resolution in Tissues, is trying to do something amazing, trying to correlate tissues, immunohistological slides analyzed with antibodies with high throughput proteomics, and is using slides from tissues and combines the visualization under the microscope or with immunoassay with high throughput proteomics analysis using mass spectrometers. And actually, he's producing a lot of data. He has produced also a lot of tools in order to do that. And he has fully focused on that the last 10 days. And they even developed artificial intelligence approaches in order to analyze this data. But during this study, he discovered once again, that there is no perfect method. So here he combined different applications of this multiplexed analysis using transcriptomics, proteomics, immunoassays. They all have cons and pros. So at the end of the day, we have to combine them and integrate their data. I think this is the current status at the moment. Another important application, this is the last one, artificial intelligence for proteomics and biomarker discovery. And this is the final scheme, collecting good quality data, molecular data, but you need to have also public repositories, text mining tools, ontologies, functional annotation for each protein. You need to have a health record for each individual and tools to integrate and visualize all of this. Visualization is also very important because if you don't have the right and good visualization, the medical doctor will never understand what is shown in this data. And this will give us some more biological insights. So the future for medicine is better personalized diagnostics, better drug target discovery, personalized drugs, if it is possible, and all of this require the integration of multiple heterogeneous data. Data, content, knowledge, understanding, wisdom. Wisdom is the clinical application. Is where we combine all this information in order to build a solution for the clinical practice. I would like to thank you all once again for the kind invitation and all the people in my lab that without them, I couldn't produce all this information and learn some more things. Thank you very much.
Thank you very much for this very interesting and comprehensive presentation. Do you have any questions? Actually, I have a question because the data you showed us are very interesting. And actually, you mentioned that with this methodology, we can even detect interactions. And I think in clinical chemistry, we're strict into identifying molecules. So I think that the future mechanistically, actually, in disease is interaction. So how feasible this could be to go, let's say, in clinical practice in the future.
Yes, this is one of the most difficult things because, as you said, you don't identify a molecule, but you try to find the lack of an interaction or the development of a new interaction. So far, this is done in preclinical analysis, and one possible application could be to have a bait, to have in the clinical practice something like an antibody, and try to, with a special conformation, and in your biological sample, try to fish the prey of this interactor. But I think this will be one of the most difficult things to do in the future. I think the first application will be using artificial intelligence to predict by the molecular identification the possible lack or presence of an interaction.
Thank you. We have a question.
Thanks very much for the nice overview about proteomics. I'm really curious about these 2,900 proteins that you identified in serum. Did you use any immunodepletion or any fractionations to achieve those numbers? And how long did it take to get those numbers?
That's a very good question because it's serum, and of course you have all these high abundant proteins. So we have decided not to deplete albumin or other high abundant proteins because at the moment we have some information that albumin is also affected in different diseases and has modifications, has alterations that it's good to see them. We managed to go so deep into the proteome because we used liquid chromatography that can separate peptides in a way that we can increase the coverage of the proteome. That was done also with a collaboration of Martina Samiotakis in Fleming. So there is a development in liquid chromatography that can go deeper in the separation of complex mixtures and can find more proteins, low abundant proteins in serum that could be masked by albumin.
Okay.
Very good question.
Thank you. So thank you all, and thank you very much.
Thank you.